hundreds of milligrams, sometimes 1,000 or more. It gets actually filtered and we excrete it. You may take an 800 milligram, let's go with Tylenol, okay? Because they tell us now, they know that Tylenol hurts the liver, right? Okay? This is why. It works so hard to filter that out of the body that by the time it's done, very little is making it to helping with the pain. So, does that make sense? It's kind of interesting, isn't it? Have you ever thought about it like that before? Michelle?
Is it the fillers that's like that it's filtering out, or like, you know, of the medication, the things that make it bind and all that stuff?
Well, now, see, you got to remember that when we have that situation, whether it's in a capsule form, whether it's in a pill form, we have it together with binders, with those fillers. And so all of it is considered foreign to the body. And so that's what the body wants to do. The body wants to filter it to get rid of it. So medications, when they talk about medications being hard on the body, medications are hard on the body. But we're still looking at something that could possibly make it to the tissue level. Well, let's go with something else, something that's going to be pretty common that you are definitely going to see a lot of when you get into the fields. Insulin. Someone who may have to take an insulin injection. Where do they normally give themselves the shot? In the abdomen, somewhere within the abdominal area. Why?
The intestines are closer to the food.
Very quick absorption. And what's the overall goal of insulin?
Increase glucose uptake of the cells.
To go and make these cells at this tissue level use the insulin so that we can have our glucose used properly. Pretty amazing stuff that happens. So when they refer to this as being the business end, that's a really good term for this, because we're getting to bring in what's considered, hopefully, the good stuff. And as those cells carry out their job, as they do what they're supposed to do, okay? Well, one of the things that happens, they'll make their product, okay? But in doing that, they're going to consume energy. That glucose molecule, they can also use fats as an energy source, okay? If they have to, which we don't want them to, they can use proteins, meaning the muscle cell, the actin and myosin of muscle cells will literally break down so that these cells can get energy. The goal is to make sure they get energy from glucose first, fats second, and hopefully never proteins. But in doing all of this, these cells produce a lot of waste. All right? So we're going to see that we have stuff, for example, like that CO2. All right? We may have some nitrogen wastes that are in those tissues. Any of the processes that the cells have to carry out in those chemical reactions they can generate waste. We need to make sure that that waste gets back to the bloodstream so that the body can filter it, get rid of it, that sort of thing, and keep the tissue and those cells, okay, which are keeping this tissue alive, keep these alive. Do you guys remember from like general bio how it talks about the cell is the living unit? Okay, if the cell is not working, this tissue is not working. If this tissue is not working, that organ's not working, that organism is not working. And this is how it does it. It does it at this capillary end. So we're going to see this in future PowerPoints, but that's okay. We'll just come back to it. Now, when we begin to look at these capillaries throughout the body, all right, we're going to find that this ability to exchange materials, in some areas, we may need gaps that are very, very tiny. In some, we may need gaps that are a little bigger. So what we're going to find is that we have 3 types. And they're going to be based on how easily materials move. So when we say a continuous capillary, this is what we're going to find in most of our tissues. We're going to see that that base, okay, the basal lamina, the base, will separate the endothelium from any of these adjacent connective tissues, all right? We will see that there are intercellular clefts. So if I'm looking at that capillary, all right, intercellular, this means between the cells of that vessel that we know as the capillary will have these clefts, these gaps, these openings, okay? Now, this is going to allow for small materials to move. In the brain, capillaries we find there, they're not going to have this. We're going to have a very good supply to the brain. We have to, okay? But the brain also has to monitor. Do you guys remember the blood-brain barrier? Okay? It's got to make sure it monitors what goes through. Now, something else that can be present are something called pericytes. Not sure if you remember those when we talked about the brain structure and the vasculature. And what this is, they actually look like this little extension that kind of almost looks like an area that will wrap around the vessel in our capillaries. What that means is that these capillaries have the ability to have these little pericytes. The reason for that, they have contractile proteins. So if those contractile proteins are present on that tiny, tiny capillary, contractile, what is that telling you that they do? They contract. What that's going to allow for is control of blood flow. Meaning, blood can flow into that capillary bed or bypass it. This is important because there is no way that what blood we have in our body can supply every capillary bed at the same time. The body has to have a way to control the blood flow. And these little contractile proteins help with that. We're going to find that we have fenestrated capillaries. This is where those endothelial cells, now when we talk about those endothelial cells, when we get to the capillary where it's only one cell layer thick, this is what we're looking at. Just cell to cell, okay? And so if I've got these clefts, in between them is a cleft. And fenestrated, these are going to have pores, meaning they'll have something bigger. It's just a tiny bit bigger. And some of them will have a membrane, but what they're going to do is allow for rapid passage of materials. All right? So the ones that are just a cleft, tiny, a little bit of a control, well, quite a bit of a controlled flow. These pores, bigger flow of materials. And then we have sinusoids. Sinus, like gaps in spaces, all right? So sinusoids, these are very irregular in their structure. They are going to be found in the liver, the bone marrow, the spleen, and a couple of our organs. Blood-filled spaces. Let's think about this, okay? When I'm at this level right here, in my blood, in my vessels, okay, I have my little red blood cells, right? Look like the little donuts, okay? Do blood cells move out into my extracellular fluid? Absolutely not. If the blood cells at this, where we're looking at all these tissues of the body, okay, if blood cells are moving out at that capillary level, like I've said, something's wrong, okay? But now, when I get to a sinusoid, that is going to be large enough for a red blood cell to move. Why do I need that to occur, for example, in my liver? Why do I need it to occur in bone marrow? Why would I need a red blood cell or a blood cell, period, to be able to move at the level of my bone marrow? Al?
Are the red blood cells created in the bone marrow?
That's where they're created. The blood cells, red blood cells, white blood cells, they're little platelets. They can just go, okay? But red blood cells, white blood cells, platelets. Well, one of the things that happens, if that's coming from the bone marrow, I've got to get it to the blood. So the vessels are going to